everybody. I'm Himla Soodyall, the Executive Officer at the Academy of Science of South Africa. And it is indeed a great privilege for me to host this month's In Conversation series with my guest, Dr. Monica Mwale. And colleagues, as you know, this month is Heritage Month, and we've had previous talks on heritage, human heritage, heritage with respect to culture during the COVID period, on how we celebrate life and death, paleontology, archaeology. And this time, we are going to be celebrating biodiversity as heritage. Many of you may be aware that this year, as part of our Science 20 commitments to the G20, which South Africa is hosting the presidency of, we are discussing the subject of climate change and well-being. And during these discussions with other academies in the G20 countries, it became very apparent that we should be focusing on the well-being on the planet, with a huge, huge focus on the environment and all living things. We are indebted to Monica for availing herself today, despite her very tight schedule, to share with us some of her experiences as a geneticist who's based at the South African National Biodiversity Institute and at the National Zoological Gardens in Pretoria. Her work focuses on molecular systematics, conservation genetics, and DNA barcoding, including applications to wildlife forensics. Now, I don't want to steal her thunder because I've also asked her to give us a brief introduction into her life as a scientist. Our paths crossed about a month ago when we were both present at a tea hosted by the British ambassador. And Monica was asked to be on a panel. And when she was asked a question of how she got into science, it was absolutely mind-boggling. And immediately I said to her, I want you to be my guest on the Heritage Symposium. And here we are today with Monica as our guest, and she will go into a few aspects of her work and her research and how her life intersects with biodiversity, etc. And please, colleagues, for those of you who are online, please use the Q&A to register any questions that you would like to ask, because towards the end of the presentation, I will invite Monica to address some of those questions. So without further monologue from me, over to you, Monica. So tell us a little bit about how you got into science, and then go on to your presentation on DNA barcoding in South Africa for biodiversity conservation. Over to you.
All right. Good morning, everyone. Thank you, Prof Himla. It's my pleasure to be here to talk to you about who I am, my journey in research as a biodiversity researcher. So the theme celebrating biodiversity as heritage is very close to my heart. I've always loved biodiversity, as a backstory of who I am. I come from a town in Zambia called Livingstone, living by the Victoria Falls next to a national park. So when Prof Himla asked me to talk about my experiences, how did I end up doing sciences? I thought just telling you the experience, the exposure that I had. Just recently, like yesterday, I just want to show you this picture. This is my primary school. So, when you talk about biodiversity, it was called Zambezi. And the Zambezi River is one of the biggest rivers. So we celebrate heritage in different ways. And my school was called Zambezi Primary School. And yesterday or the day before, elephants put down that wall. So here you have people living together with nature. When I was growing up, we never saw elephants this close to our school. But now this was an image from alumni, how these elephants came into the school and just broke down the fence. So telling us that we need to manage our biodiversity, people, and we have to live together, I think, in an ecosystem that supports both humans as well as wildlife. So coming to my career, I have always loved sciences. And when I was in high school, I wanted to do science as a major at university. But I went to a school where there were no science teachers in my grade 11 and grade 12. So when we're now applying for universities, how do you fill out your forms the year before to apply for university placement? We were all told not to apply for science, apply for social sciences or other departments. But I felt it in my heart, I've always wanted to be a biologist or a zoologist. So the first form they gave me, I filled it in and I applied for natural sciences. So the career person then was very upset. She tore up the forms and said, "You are not allowed to apply for science. Apply for other courses because you don't have a teacher, you won't pass." So I filled it again. She tore it up until she ran out of forms and she was like, "You know what, Monica, I give up. You just apply, you won't make it to university." But here we go. I did make it to university. I did manage to pass sciences because my mom bought me a textbook. I studied because I really knew what I wanted to do. So science was always a passion, and that's how I ended up then doing a master's in fisheries, a PhD in systematics, looking at biodiversity, including genetics, and just studying life. I've always loved studying nature in all its forms. So this is just my brief story of how I came from this primary school now, that's going through this, to becoming a biodiversity researcher at SANBI. So biodiversity, and when you talk about our heritage, South Africa is one of the most biodiverse countries, occupying a small portion, 2% of the world's land, but home to more than 10% of our plants. 7% of all world birds and reptiles occur in South Africa. 15% of all marine species in the world can be found here. 9 biomes declared as a biodiversity hotspot, celebrating this amazing diversity of species at the southernmost tip of the world, like towards the south of the continents of the world. So this is an amazing biodiversity or heritage that we have. However, there is a loss of biodiversity as well as biological annihilation. People are moving into places. The ecosystem systems are being broken down due to anthropogenic effects. And then when you look at what we have, we actually don't even know the extent of how diverse the world is. We might be losing species more faster than we can even describe them. So a recent biodiversity assessment done by SANBI, looking at different ecosystems, looking at species, looking at the levels of how are we protecting these ecosystems, how are we protecting these species. So the NBA found that rivers, wetlands, estuarine ecosystems are the most threatened. And the protection level is not that high as well. We don't have most of these systems because most of us want to live by the sea, by the river. So there is this competition for resources. So almost 99% of estuarines or wetlands are threatened. They are moderately protected, and they have more than 2,019 species that were assessed. And 12% of these are threatened with extinction. If you look at the Convention on Biological Diversity and the targets on how to sustainably use or protect biodiversity, most of them are to halt extinction of threatened species. How do we ensure sustainable and safe and legal trade? How do we manage our wild species? How do we report on how humans are living with biodiversity and sustainability? All of these are questions or research questions that need solutions. So we need to come up with outcomes or solutions that respond to these challenges, that are aligned with strategic priorities, and these are the major ones: habitat loss, pollution, invasive species, climate change, illegal wildlife trade, and sustainable resource use. Over-exploitation of resources is all threatening this heritage that we've inherited from Mother Earth. How then do we study this or come up with solutions to advise? So my work looks at genetic analysis tools, therefore, to assess, identify biodiversity, species, genetic level, to advise government to come up with management plans for biodiversity conservation. So the true number of species on Earth is unknown. On 80% of species remain to be described. So how do we then describe this diversity? And how then do we protect these ecosystems? So in South Africa, currently at SANBI, we are involved in what is known as the South African node of the Barcode of Life project, where we are using DNA sequences, which are called DNA barcodes, to do species discovery, to understand species interactions, to track dynamics of species within ecosystems. Our aim is to describe as many species as we can and to fill in the gaps of all the missing species numbers or details of ecosystems in our threatened ecosystems in South Africa. So this project has funding through the DSTI, where there is funding under what is known as the Foundation Biodiversity Information Program to manage, to generate, and disseminate foundation information and knowledge to support decision making. And SANBI is one of the key players there within this program, advising the NRF through the DSTI to fund projects or research that contributes to these activities, to ensure that we understand what biodiversity we have locally. So my talk then will just look at where we are at the moment with within this program, what are the gaps, how can we use this genetics to study species, applications including forensics, meta-barcoding, and looking at disease vectors. So how we're starting to implement this program is to work with national checklists. We're looking at available checklists of all groups in South Africa, animals and plants. Then we're combining this with genetic data that is in databases, GenBank, and including board databases. What is out there? What information do we have on the genomes and the diversity of these groups? And then to combining those data sets to find where the gaps are in the data. The gaps are in terms of ecosystems, in terms of area, in terms of capacity and institutional support to ensure that we can do this. So currently, these are the current checklists that we're working for. So SANBI hosts the national checklists. We work with experts locally, including internationally, when we don't have local experts, to understand how the diversity or to come up with a number of species in South Africa. So these are the groups that we had that we're looking for. There are a lot more, but there is just limited expertise to come up with these checklists. So the first group I'll look at is mammals. Everybody knows mammals. We all have mammals. Most of the charismatic species are mammals: elephant, rhino, lion. Almost everything that everybody loves or studies is within mammals. So how are we doing there? So if you look at the current of species that we have assessed in South Africa, there's 336, but 57 of those species are endemic to South Africa. And if you look at the numbers or the percentage of least concern, we have about 30% of our species that are actually in the threatened categories. Then when you look closer at mammals, small mammals are so underrepresented. There's very little work that's been done on them. So even for a group of that is charismatic like mammals, we still have huge gaps in our knowledge of the diversity. The next one is reptiles. Again, 45% of all reptiles in South Africa are endemic. So this is a huge heritage. 45% of our species only are found here. But then if you look at the diversity, 9 species that are highly traded, listed on CITES, do not even have DNA barcodes for us to do delimitation and study and understand the diversity. So there is, again, a huge gap. So there's limited information on species, be it taxonomic. We don't even know what the distribution ranges of most of these species are. If you look at this more closely, looking at one of the orders of Reptilia, squamata, we have 32 listed on CITES. Once you're listed on CITES, it means trade threatens the survival of that species in the wild. But if you look at the gaps, we have 199 non-endemic species, 156 are missing. So there is a huge gap again in us setting up or building these species reference databases using barcoding. Another group is elasmobranchs or your sharks. If you look at the diversity of that locally in South Africa, we are one of the 5 endemic hotspots globally. So a huge heritage, huge diversity, ecosystems that are so diverse and unique and are only found in this area of the world. So if you look at the number of recorded species, over 205, but if you look at records of species that we have added to this database. 128 of those species are missing on the Barcode of Life database, which is like a program we want to set up for species identification. So there's still a lot of work still to be done, and always there's more and more species being described, new sharks being described. So there's 5 non-endemics. Some of them have been sequenced, but still the data is low for all species. So again, we don't have data on our diversity. If you look at species that do not even have one record, there are over 36 species that do not even have a record on this database that we're building. So there's need to study our diversity and understand these species before we lose them. Then another thing which we are seeing as even the things that we think we know, they are more diverse than we think they are. So this was a master's student project I had with one of my students, where there's this species known as the Gymnothorax undulatus, widespread species found from South Africa all the way to the Indo-Pacific. We thought it was one species. During this master's project, we found that what we called undulatus in South Africa, this green clade, is very different from what is the widespread form. So if you look at the genetics, these are other critical key other species which have distinct morphological features that cluster with this lineage from South Africa of what we thought was a widespread species. So on one hand, we have species we don't have on the database. On the other hand, we have species we think we have on the database, but actually they're more diverse than we think. We think they are and could be representing different species. Cool. Then when it comes to forensics, so building databases to support forensics is becoming more and more critical. As more and more critical species are traded, we need to be able to identify what's involved in the illegal wildlife trade. So we are doing a project together with the police, the National Prosecuting Authority, and funded by UNEP to come to set up reference databases that enhance forensic capacity locally. So in this project, we are working with the police, environmental affairs, National Prosecuting Authority. We have got 3 institutions, University of Johannesburg doing plants, SANBI is doing mammals, reptiles, and birds, and then SAIA, which is another facility of the NRF, is working at aquatic biodiversity. And through this, we are setting up reference databases to support species identification. We are setting up standard operating procedures and guidelines on how to do this forensic casework effectively so that the results we produce will be accurate, reliable, and can be used in court, like they'll be fit for purpose for prosecution. So we are working in line with the South African National Integrated Strategy to combat wildlife trafficking. We've identified the key species involved in trafficking that are highly listed among birds. So we're looking at about 372 species. Our list, if you include plants and other organisms, is about 900 species that we need to barcode. All of this is to support enforcement. What we have done as well is within this database, we're ensuring that we're including all lookalikes. So for example, when we work on the big cats, we have jaguar DNA, we have tiger DNA. They're not endemic to South Africa or indigenous, to South Africa, but it's important for us to differentiate our species from similarly looking alike species. So the cheetah, the leopard, and the jaguar all have to be in this database. Because beyond us having indigenous species, we also have businesses that are breeding exotic species. So we need to be able to identify all species that are found within our borders. So this is to enable appropriate enforcement actions. So how far have we gone? If you look at the mammals, we've included for the cats, we've included all the different cats that you find in South Africa. So we've got civet, black-footed cat, tiger, lion, and you can see that our database can discriminate between these species. So why is it important to do this? Mainly in illegal trade, what you find is processed body parts. You might find lion bones or bones of a tiger. Sometimes you just have hair or fur, and you will not be able to identify. The police may not be able to identify that species. So then we use those biological materials and do genetic testing and associate those to a species that's in a database. So far we know our database works and there's good species differentiation. Other things like for lizards, I don't know if you've heard of the girdled lizards or the sun gazer. It's in a genus called smog, like the dragon in the Lord of the Rings. So this is also highly traded to Europe. So we've had to add those species within that group into our database to support forensic enforcement. So at present, what have we done? So in terms of forensic case work, we've done over 700 police cases for the police. And most of the questions that we answer for the police is, what species is it? Who is this specimen? Have we found a suspect with a sample that we can link to a crime scene in Kruger National Park or in some captive facility? So when we say who, can we get sample from a crime scene and link it to something that's in the border, crossing the border? Or where is it come from? Can we say this species or this poaching activity is happening in Kruger, in the Eastern Cape, or in the Northwest Cape? So we need to know traceability to support the police so then they can manage those illegal trade routes. So an example of a case that we did was on shark fin confiscation, a huge consignment that was caught in Cape Town, and we had over 175 samples we took from those sharks, and we suspected that they were selling sharks that are listed on CITES. So when we analyzed, we found hammerhead sharks, and hammerhead sharks are listed as on CITES Appendix 1, and we are not allowed to catch or sell. The whole genus is listed. So we found hammerhead sharks in this consignment of shark fin. So this was a conviction. However, these are big international corporations that would just pay the fine the same day. So in the fight against crime, I think we need to find ways in how we can protect our heritage, our species, but ensure that prosecution also protects the species. So another work that we're doing again with barcoding is understanding pathogens or vectors of disease. How do we protect our species with these disease outbreaks? Do we know, like for ticks and other vectors, what's their diversity in South Africa? Where can we find them? So we had a project where we did a gap analysis on ticks, focusing on tick-borne pathogens. How many tick species can we identify? And even in that paper that we did, there's so much diversity, but there's so little information out there. We have very limited expertise. We do not even, for most of these species, 50 to 100% of these groups have no genetic reference samples that we can use to identify them. Either not for invasive species monitoring or to monitor the spread of disease within our communities or within our national parks. Another example is the work we're doing in national reserves. Like there is a need to have non-invasive sampling of environments. So we're using environmental DNA to understand diversity in national parks, in reserves, in cities, in towns. So we've been like in Pretoria to Ferregrain Reserve, where people go to see what's the diversity within that river system that goes within the reserve. And in this paper, we looked at a Bosanali Reserve in the Northwest, where there is they know, I think they've got over 100 or something species known from that reserve, but there's some cryptic species they're not aware of that might occur in their reserve. So we did an eDNA study with them. This work is still undergo, but we analyzed the water, the sediment, to see whether their own mark and capture methods of monitoring diversity will equate to what we're going to do with the eDNA. So this is the potential, and right now we've been invited also to Kruger National Park to do the same, to understand the diversity of species across Kruger, to also get water from water holes, to know how these animals are assembling within the reserve, because that allows management to manage them much more effectively. So all of this is possible because we can do in-DNA analysis by matching our DNA samples to the DNA barcoding database. So you can see there is so much that we do. This is my final slide, but I thought I'm giving you a snapshot of the work that I do and how, it's all connected, how it links back to the ecosystem, to genetic diversity, and to species. So my final thoughts is we do have very diverse taxa. There's so much work to do in terms of our biodiversity heritage. How do we understand what we have, how to protect it, where it occurs, how it's distributed, and how it's in contact with other ecosystems and people. So we are finding that there's so much diversity, cryptic diversity. We are also finding geographic variation. Therefore, if somebody wants to have a big project in something like the Karoo, can we use that space to build something if we think that species is widespread? But in most cases, you need to know the geographic variation because you might be getting rid of a unique variant of a species in an area, even if it's widespread. So understanding that allows us to make good decisions for sustainable use and for management of species. So we're finding that the work that's being done by morphologists seems to match the genetic diversity, so we're happy with that. And this therefore supports forensic applications. As you have seen, we've used barcoding in a number of cases to support the South African Police Service. And an important thing, though, is the availability of museum collections and samples to do this work. So beyond us just doing barcoding, the capacity to collect the samples, to identify them, is critical. And that's where we're partnering with museums. In some cases, we are going to historical samples of specimens that we are unable to access and barcoding those ones for the database. So the value of collections is becoming more and more important for us. And then using new next generation sequencing technologies and others to monitor systems, disease, as well as to understand diversity of diets and ecosystem. All of these respond and can use the DNA barcoding database to answer to answer those questions. So yes, that's my talk. And I would like to thank you for the invitation and organizing the webinar. And these are just some of the colleagues I'm acknowledging in this talk. Thank you.
Wow, thank you. Absolutely fascinating. Apologies, I'm not going to switch on my camera because it already crashed once. So hopefully this way it doesn't, but I will switch it on a little bit later. So just to confirm, Monica, what are you sequencing when you do the barcode? Is it the 12S, the ribosomal sequence?
So we have different gene regions for different taxon groups. So
Okay.
for mammals and other vertebrates, we use CO1 and 12S and
Okay.
16S, including cytochrome B. So what we found is different genes have different mutation rates. So we use a specific gene for a specific group.
So for the non-genetic specialists, could you perhaps explain why those regions are selected for the sequencing? And the second part to that question is there a universal agreement in which regions to sequence so that the comparison across different geographic regions are compatible?
Okay, so there is the Africa. I'll start, I think, with IBO. So barcoding is managed through the International Barcode of Life initiative. So it's got several working groups of experts in genetics and taxonomy that are working on various organisms or phyla. So, for example, you've got a vertebrate working group, you've got one working on protists or bacteria, you've got one working on plants. So what they try and find is you want to find a gene region. So when you talk about barcoding, you are literally looking at a gene region that has got high interspecific diversity. I hope for non-geneticists, where there's enough variation between species, but then there's low intraspecific variation. So all organisms have variation within each other, but you want variation within a population of a species to be lower than the variation between a species and another species. So there is something that we do which we call the barcoding gap analysis. You would like to find a gap between intraspecific variation within a species versus comparing that species to another species. So, for
Right.
example, yeah, I don't know if that's
So go,
clear.
go, go, go ahead. Go ahead.
Yes. For example, for the CO1 gene, when we're looking at things like fishes, we have found that the variation within species or populations is generally between 0 to 2% for the gene region that we're looking at. So 2% mutations between individuals that belong to the same species. Then when we go above 2%, those comparisons become comparisons to congenial or closely related species. So therefore, the cutoff point for CO1 is 2%. Anything above 2% variation will be clustered into another group and as a potential species.
Okay.
So again, this all depends on the groups you are working on and the specific gene region. So for reptiles, for example, this is a long-lived group of organisms that have lived for millions of years, accumulated a lot of mutations and are so diverse. So you would find even within a species higher percentages, up to 5% between individuals of a species. So yeah, it depends on the group you are working on, and you have to have enough data to come up with these what we call thresholds to define intraspecific within a species as opposed to interspecific.
Right, right. So, I mean, the reptile story is also very fascinating because there's a very strong link to the dinosaurs with reptiles. You want to share a little bit of your thoughts on that?
Yes, I think when you look at reptiles, there was periods I think in their evolution where there was some radiation in some groups, but these are organisms that have in most cases have found a niche earlier on and then have survived in that niche without needs to adapt further. So over time they've kept on accumulating mutations, but it's the same form. For example, you
Right.
look at crocodiles. There have been crocodiles. I think because they've adapted to a niche and an environment where there's not too much competition. So they're sort of they're not adapting to find other environments quickly. So they accumulate mutations over time, over a long period, without changing drastically into other new lineages of forms. So yes, so some long-lived groups that have been here for millions of years who have accumulated a lot more mutations. For example, versus Lake Malawi cichlids. I don't know if
Yes.
you're all familiar with Lake Malawi. I think was filled in the Pleistocene about
Yeah.
2 million years ago. So these species that were found in there then rapidly evolved and underwent this adaptive radiation. And when you look at the CO1 gene, the one I was talking about, there's no
Yes.
difference between species in Lake
Wow.
Malawi. They are all less than
Amazing.
2% divergence. You
Yeah.
cannot separate them using the CO1 gene.
Right, right.
Yeah.
Colleagues, for those of you who want to ask questions, please write in the chat. And if you want to be online, just raise your hand, and my colleagues monitoring in the background will beam you up into the existing chat to ask your question. Monica, fish species tend to be a very good species to study human gene homologs, that is, genes that are found in the fish but are also in the human. So if you want to study them further, they use some of the, like the zebrafish
Zebrafish.
and other ones. Can you share some of that knowledge with us? Are you aware of those types of studies?
Not really. I'm not aware of it. The only thing is, oh yeah, I think I told you I did fisheries. What I found, okay, I'll just tell you my own interest in why I like fishes. Every life form, every behavior can be found under the sea. You find,
Right.
like, for my PhD, I worked on pipe fishes,
Yes.
and pipe fishes are like seahorses in the males getting pregnant. So
Hmm.
the female transfers her gravid eggs to a male who broods them, fertilizes them and broods them, and actually has contractions and gives birth. And then some
Right.
species have parental care, some do not. So if you look at humans and different types of behavior, then I also worked on a group of fishes that have an antifreeze protein that occur in the subantarctic. So you have got
Right.
these species that occur from Antarctica, but they've got this antifreeze protein that lets them live at sub-zero temperature. And the level of expression changes. So if they move closer
Right.
to the subantarctic, they have got less antifreeze protein. If they move closer to Antarctica, they have more, what do you call it? Yeah. So I think fishes are so diverse. You find fishes with a parasitic male that spends all its life on the female.
Right. Right.
Um, you also get a lot of these line fishes. I don't know if you're familiar with South African line fishing and fishes. There you have females that change sex and become males. So
You.
if they are in a school of fish and the male gets taken out because it's the bigger individual, the largest female changes its sex and becomes the male. So it's like every pattern of behavior you can think of.
Fascinating.
Um,
Fascinating.
and
Yeah.
then you have got mimics in the sea,
Yes.
fish that look exactly. So you have like these fishes, the cleaner fishes. I think if you've watched Shark Tale, have you seen the cleaner fishes and the cleaner shrimp that clean up all the other fish?
Yeah.
But then you've got these fang blennies that mimic them, look like a
Right.
cleaner fish, go to a cleaning station just to take food, like to take a bit of tissue or a scale or suck blood. So it's like
Yes.
every weird behavior you can think of. So I probably that's why they would study them. I don't know, but I'm not familiar with the homologs and
Yes.
human.
No. The study that comes to mind, and I forget the details because it's been some time ago now. But some of the pigmentation genes responsible for color, a group of scientists were using, I think it was zebrafish, as the system to study whatever they were studying. And they accidentally happened upon a mutation that changed, the stripes to non-striped fish. And when they
Wow.
looked at it a little bit further, they found the homologue in the human genome, and further studies on the human side revealed a cluster of genes. I forget the specific names, etc. As I say, it's been almost 15, 20 years that the study was
Yes.
done. And hence the fascination of how you can use other species to study human genes, and that just reinforces the sequence of evolution. We talk about Luca, the last universal common ancestor, being a single cell from which the radiation from prokaryotes to eukaryotes to mammals, etc., have evolved. And that is why there are similarities of genes across species for particular functions. But there is a guest, Augustine Niba, who's in the chat. Augustine, over to you to ask your question or to your comment. Over to you.
Thank you. Hi, Monica.
Hello.
That's a beautiful, beautiful presentation there. And
Thank
that
you.
leaves me with a lot of questions. I'm working on invertebrates, and you know how numerous they are. It's a whole big pack. How do you find a way around that in terms of taxonomists and do you how is the way forward about that group of animals?
All right. So thank you for that question. So at the moment, we've got a program, a project that's looking at macroinvertebrates. I don't know if you're familiar with that for freshwater. I'll start with freshwater. How they use microinvertebrates to monitor freshwater systems, the quality of the ecosystems. So they're using that SARS monitoring. But what we have started doing now is using eDNA and actually barcoding macroinvertebrates in these threatened water systems. So I've got a PhD who's looking at the waterbed, uh, wetlands there, where we've collected macroinvertebrates from the upper reaches to the lower reaches just to see the diversity. We have also collected water to check the heavy metal, water quality issues. But I can tell you now, when we're sequencing these, there are very few references for us to match them. So our data sets will potentially be will grow. We have to grow the databases for barcoding to support monitoring. There's very limited expertise in macroinvertebrate monitoring. So in Sandy, we had a project where we are looking at fresh water. It's called Refresh. And I think on that group, we just have maybe 4 or 3 experts that work on taxonomy and actually studying diversity of macroinvertebrates. So we have a strategy in place to study more macroinvertebrates and improve our knowledge of them because they're important. I mean, if we can use them to monitor ecosystems, we need to know how diverse they are and what we have. So that's for freshwater. For marine, we have a program with SCION. I don't know if you know the South African Environmental Marine, where we are looking at deep sea invertebrates. So with marine studies, we know almost everything up to 35 meters, but when you start going deeper, we don't even know much of what diversity there is. And there's potentially a lot of species that we have not described or we are not even aware of that occur in the marine systems. So we have been working with SCION. We have got a project called CMAP, trying to map the diversity of the oceans. And these are invertebrates that come up in trawls or in other researches that we are then barcoding or sequencing. So yes, there's a lot happening with invertebrates, with the invertebrates like your insects. We've had projects with Malay straps where we're looking at diversity in Kruger National Park, in other areas. We also put an interesting project we did. We put traps in all the national botanical gardens to find out what the diversity is in these areas where people are coming in contact with nature and with wildlife. So yes, we're trying to increase our database, and yes, that's a huge, huge gap for us. I hope I've answered.
Especially, I mean, especially the terrestrial ones, the terrestrial ones.
Terrestrial ones, yes.
Terrestrial ones are actually the ones I'm really interested in. Yeah. Thank you very much.
Okay.
Wonderful.
Now you can, I'll put my details. I think then you can email me if you want me to link up with others. But yes, we are looking for partners. We are looking for teams to support this. Yes.
Thank you.
Excellent, excellent. There are a few questions in the chat. I will get to that, but I just want to pursue the barcoding as a way of identifying illegal trading procedures. So, I mean, we know rhinos are being threatened, for example. Pangolins are another problem. So how does that process work? And what is your timeline in order to, because, you know, these things go very quickly over the borders and in ships, etc. So how does the barcoding system and the system that is in place to do this kind of work come into play in the real world? That is your world.
Okay. Yes. So what happens is with, for example, rhino poaching, the poaching will happen on a private reserve or it will happen in a national park or when the police are doing their traffic controls, then they come across somebody who's carrying, it's got contraband. So that's how it does. Or they could be investigating, somebody that's been investigating over years, suspicion of trading in rhino, and they're trying to investigate this case. So what comes to us will always be an investigation question like, can you please identify the species involved in this crime? Or can you match these species to a crime that happened this on to the horns we've given you from these 5 places?
Okay.
So for ongoing investigations, the investigator tries to build a case. They'll give us samples and they say, add to the database, add to the database, add to the database. Then once they apprehend someone for a rhino, they'll say, dear Monica, can you please now match this to all those samples I gave you from Kruger? Because now they want to identify where that crime happened or the place where this happened. So that's what usually happens with the rhino, or it could be at the border. If a person is captured at the border and you want to check what he's carrying. So the law, in order to prosecute someone in South Africa, we work with the TOPS, the threatened or protected species. If a species is listed, we can prosecute someone. If a species is listed on CITES, you need a CITES permit to possess that species. So a crime is committed if you do not have a permit to possess that organism or that. So that's how you can try those people. So we get all sorts of things. If it's a crime scene, we can get panga or whatever was used to kill the animal. In some cases, people have gone to a crime scene and have brought us a soil sample with suspected blood on it. And then we check whether that blood is from an animal, whether that blood then is a rhino or an elephant. So yes, so
Right.
that's how it happens. Then for pangolin, it's the same as well. The police have been working, so they are suspicious. They know, they are sort of familiar with what the routes are for trafficking. They get tip-offs, or they are actually investigating specific people. And in most cases, it's those, or they just find the consignment at a border post trying to
Right.
leave the country. Yeah.
Right.
So
And
I
then,
can give you an example. Yeah.
yeah, what is the biggest bottleneck in turning lab evidence into courtroom-ready cases?
The biggest bottleneck is mistakes that happen in the process. So
Ah,
defense do not attack the DNA. They do not attack the DNA, but they will attack and say the label is different. The label from the crime scene had a 7. The label from the lab says ends with a 6. So those small errors you can make in the system is what they go for. Or they'll say the process of the lab is unclear. Can we see your SOP? And they'll go for your SOP, but your SOP says this, but this is what you're giving us. So they rarely go for the DNA result. They'll go
yeah,
for the process. They'll try and find a mistake in the process so that they can throw out the case.
the case, yeah. There's a
So
question.
reporting accurately, writing the correct thing, affidavits must be clear, simple, with no cause for, yeah.
Ambiguity and things like that. Yes, yeah. The one of the questions on the chat is, what role is or can be played by traditional and religious organizations in protecting our biodiversity? And then, according to your opinion, are these groupings doing enough? That is the
Okay.
traditional and religious organizations.
Okay. So traditional and religious organizations play a big role. They've been the custodians of this biodiversity for centuries, for years. So they need to be included as part of the process in making decisions to protect the species in these communities. I think for a long time, it's been a top-down approach of why,
Yes,
finding out why these communities are getting involved in the illegal crime. I think from the beginning is because they feel they are left out. They were never part of the process. All of a sudden, there's a fence near them. They can't go where they've hunted for generations. For example, my grandfather was a hunter. He used to hunt for his village. His role was to provide meat for the village. So I
right.
come from that background where my grandfather, if I go to the village, they'll say the daughter of the hunter is here. Because that was our historical, that's where
Right.
I come from. So I think they play a big role. And as we continue to engage with communities, we find that they actually understand in most cases what's going on in those systems, why these animals are behaving the way they are, and they could actually come back with better solutions on how to protect these organisms. For example, I think an example is like with the pangolins. Pangolins are regarded as sacred animals in
Mm,
Limpopo province. I mean, if you kill one, I think they say you have drought. So there's culture and there's belief systems associated with these species. But the
right.
problem is, I think there's that breakdown. Young people are unemployed. There are people coming into communities and offering big amounts of money. Systems, probably community systems or the cultural system is breaking down. But if we start involving them as leaders, I think society listens to the leaders and we can transform those communities. And we've got had projects like in KZN where they now have these um, communities where they are working together with reserves to manage the wildlife, employing the people from the community, building schools in those communities, building hospital and other infrastructures so that those communities can thrive without,
Yes.
yes. Because we always say these communities have lived for years with this wildlife and there were no problems. But in the past 30 years, what has changed? So yes, they have to be involved. No more top. It has to be bottom up. All decisions
Yeah.
have to be bottom up. Yeah.
And then the two working together, buffering circumstantial conditions versus the
Yes.
protection protocol. And then, of course, you've highlighted already belief systems, etc., and myths. There's indigenous knowledge and mythology
Yes.
going after particular animals, etc. And I suppose with more discussions and more open dialogue, there would be a better buffering of the two. There's one question in the chat. This is a little bit of a tricky one, but I'll see how you can handle this one. Trying to follow the incidence of virus species integrations and mutual evolutionary DNA transformations. The person asking the question is just wondering if any of this is relevant to what you are doing. So when you have virus and species integrate into genomes from an evolutionary perspective, have you encountered that? Or is that of interest to you as well? I hope I
Yeah.
understand the
Sorry, what is
question
the
right.
question? Yes. Is it one of the
Yeah,
in the chats?
it's
I'm trying
it's
to
in the
read it.
chat. It's asked by Hester Duplessis.
Oh, okay. Oh, yeah. Trying to follow incidents of virus, species
Yeah,
integrations, and mutual evolution, DNA transformation. I just wondered if this is relevant to you. Okay, so I'm not that familiar with them, but we do find something in the barcodes. So
right.
there is these things called NAMPs, nuclear
Yes,
mitochondrial into the that's go. I
correct.
think what's it called? NAMPs. Yeah. So sometimes when we sequence for the cats, I'll tell you what happens. So we're sequencing tiger, lion, and cheetah, but we're sequencing the mitochondrial genome that's
Right,
been integrated into the cat genome. So
right.
what we were finding was this leopard is a tiger, this tiger is a is a leopard, just because of this. But when you sequence that genome, it's not the
Yeah,
full genome because it's an integrated one. So we
correct.
call them NAMPs. So what we had to do is design specific primers, more specific, to amplify the correct mitochondrial genome for
Yes.
the cats. So yes, we are finding that a lot. Another example is I worked with falcons when we, because falcons are traded. They've been used for centuries in sport and everything. So they're important also to monitor. So when we're creating that, we're finding this deletion of like 10 base pairs within the CO1 gene, which is not supposed to happen in mitochondrial
Yeah,
DNA. So we knew that was then we are sequencing a NAMPT, if
right.
something, yeah, a mitochondrial genome that was not, yes. But over
Mm,
time has also accumulated mutations, but it's not the genome. So yes,
yes,
we found them in birds and we found them in felids, the
yes.
large cats.
And from an evolutionary perspective, lines and signs, lines standing for long integrated nuclear elements and signs short
Yes,
integrated nuclear elements, does create, differences in lengths of particular areas on the genome. And lines
yes.
and signs have served as a very, very good way to study for example, human evolution. And I'm sure the same must be the case for the animal kingdom. But that's a different kind of lecture. That is why I said it's a little bit of a tricky question.
It is tricky,
There's
yeah.
a very nice one, given that we are talking about heritage and celebrating Heritage Month. And the question is, what does Heritage Month mean to you wearing your environmental lens?
I think Heritage Month, wearing my environmental lens means I'm living in an environment where the ecosystem is functioning, where I can live at one with nature. I think
Yes.
I love my favorite moments is when I go to the ocean. I love the sea. So heritage means to me is a clean space, a clean environment where humans living at peace with nature. So That is heritage. Beyond that is the cultural heritage stuff, but environmental heritage means seeing the places where I've grown up. I showed you that first slide of my
Yes.
school. That never used to happen. I cannot believe elephants are now coming into the schools. So what has happened? I never knew that I lived next to a national park without a fence that was less than 10 kilometers from me. But lions
Yeah.
never came where we were. Elephants never came near us. We never had this. And I used to walk at night to school. But now, the elephants are coming into the towns. We're getting lions. We're getting, it's like the ecosystem is not balanced. So heritage to me means looking after what we've received and ensuring that it's there for future generations. I love the environment. I love biodiversity. I love nature. And I would like to protect this heritage, which I think is for all of us.
Yes.
That's how I am.
No, you're absolutely right. I mean, the human intervention has encroached onto the biodiversity space, threatening the plant diversity and spaces where they can populate.
Yeah,
Similarly, it happens for animals, whereas in the past, animals roam freely, and as humans have encroached into their territories, obviously there's going to be a clash or a
yeah,
competition for that resource and that space. I mean, if you
yeah,
go to St. Lucia, at night the hippos come out of the water and they go visiting onto the streets, and we aware of those kinds of stories. And as you
yes,
mentioned about elephants coming in, um, as their ecosystem gets dwindling, they are going to traverse the more human populated areas.
yeah,
And we in South Africa are very privileged that we still have reserves and laws protecting environmental spaces, etc., so that
yeah,
we can at least preserve those ecosystems for future generations to come.
yeah.
And I know we're going on in time, and I don't want to take you away too much longer from your work, Monica. But again, as you can see, there's a huge interest in this particular topic. You've shared with us the kind of meaning that we as a species can learn from to encourage the living spaces of other living things. My last question, in keeping with our S20 theme of climate change and well-being, is can you maybe make a comment on how climate change affects the well-being not just of the planet and the environment, but of all living things? And I'll let you close on that comment. Over.
All right, thank you. I think climate change in the past few years has shown us the differences. You've seen how the sea, these cities have been affected with these huge waves. I think I've got a professor I work with. His project is called Hot Birds. In Australia and in the Karoo, these temperatures going over 50, birds are literally falling from the sky because of how hot it is. Falling down like completely, these birds are not able to move because they cannot cope. So we have also seen changes in the way organisms are behaving. For example, I don't know if you're familiar with the sardine run, where we have the sardines going up the coast. And I think if you have noticed, that's changed. It's no longer working the way it used to. So you
Yeah.
have got all these industries, all these people who have built their businesses on doing these sardine runs. Some years it's not the way it was. There is also now instances of the great white sharks not being in our oceans very as much as they used to be. We've had an extinction of the northern white rhino, of the south,
Mhm.
yeah, of the yes. So climate change. I think for those who are not familiar, there always is climate change with glaciations and over time and temperatures go up, there's cooling and what. But due to anthropogenic factors, it's happening faster than it's supposed to happen. And historically, or if we look at our evolution, animals have time to adapt to those changes, and then they move into different environments, distributions will shift and move into new environments, but this is happening too fast. I'll give you an example of my PhD. I said I worked on the pipefish. So there is this estuary where the pipefishes occur, but they need fresh water coming in. And this estuary is temporarily closed and it opens every so often, every 2 years it opens. But now people have built their houses by the estuary. So at night, because they can't wait now for it to open naturally, they go and bulldoze it open. And then I was called to rush and collect. There was a fish queue because the fishes were not ready for the opening. This happens. So there was this fish queue on the reeds in the base, a lot of dead fishes because this is not how it's supposed to happen. And most of the samples I analyzed for genetics came from that fish queue because there's nothing that we could do. So yes, climate change is happening, it's here, but we need to find ways in how to make our ecosystems more resilient. People need to start responding and make more resilient so we don't need to damage those ecosystems that have been protecting us. And as we wear them down, there's no more resilience in the system to manage these effects of rapidly happening or escalating climate change. So it's resilience. We are losing the resilience of ecosystems.
Yeah, no, I love the choice of your words and the way in which you passionately speak about things. Colleagues, time is up, and with somebody who's so passionate about her work as Monica is, we could go on forever and ever. And you can see why when I heard her speak the other day, I absolutely had to have her on this platform to share with us her insights. And you have just done justice to the theme of celebrating biodiversity as part of our heritage. So thank you very, very much for that. And I just want to give a shout out to all the naturalists and conservation specialists out there who work tirelessly to protect our environment, to share with us their journeys and expertise. I'm a huge, huge wildlife fanatic. I start my day with the program and I kind of end it. And if I don't watch it, I feel like something is missing in my life. And so I just want to acknowledge the people who are part and parcel of that ecosystem of preserving our environment for future generations to enjoy the kinds of biodiversity that is currently being threatened, but hopefully will go on into the future. Thank you all for joining us. This conversation has been recorded and will be shared on our Facebook and YouTube platforms. So please pass it on to others who are not available today during this time slot to watch it, because it will be really, really insightful, particularly for young people who are passionate about the environment. I think you did justice in explaining some of the very complex, thoughts and ideas to us. So to one and all from us at the Academy, I want to wish you all a very happy Heritage Day next week, and continue the spirit of preserving our environment, not only during Heritage Month, but throughout. So thank you all, and thanks to my colleagues for helping support everything in the background. Have a good day forward. Until we meet again, all the best. Bye for now. Ciao.
Bye. Thank you. Thank you.